රංජනි ඔක්කොම හරි අර අතන යටින් අර වැටෙනව නේද stop sharing කියලා. ඒකේ මෙහා පැත්තේ hide කියලා තියෙන්නේ. ඒක hide කරන්න නැත්නම් උතන touch වුණොත් ඔක share වෙන එක නවතින්නේ. ඔය hide කරන්න.
Good morning, everybody. I welcome you all on behalf of the Anti Society of Medicine for the CMB lecture. Crowds are this, but we are doing a live webcast to other hospitals, Peradeniya, Mahatale and Navalapitiya. Interested parties also can join individually from home or mobile. And also this lecture will be available in the YouTube as a recording for people who are interested. Coming to the lecture, I think today's speaker is no outsider to our society. Prof.Kalana Maduwage. He graduated from Peradeniya Medical Faculty. Did his M.Phil in Sri Lanka, PhD Australia, and FRSPH in UK. Currently, Professor Kalana is attached to the Biochemistry department of the Peradeniya University. Today's topic is therapeutic use of nature's deadliest weapon, Venom. He is the best person to talk on this because he has done many research about snakes and venom. He has a keen interest, I think few of us, we all know about it. He has a keen interest in reptiles and freshwater fish. He has done a lot of research and identified 15 new species of freshwater fish in Sri Lanka, and also 3 species of snakes and 1 lizard species. So altogether he has contributed 19 species to the faunal list of Sri Lanka. He also has invented a new test which is recently they started practicing it at Peradeniya. You can test the venom concentration in blood and also the type of species. So that is also a new invention. So I call upon Professor Kalana to deliver the lecture.
Good morning to all of you. Thank you very much, Dr. Kalana Maduwage, for inviting me for this lecture. This topic may not be directly related to your postgraduate studies or your clinical practice day-to-day that you are doing in the ward setup. But I thought to discuss about this because there may be some interested medical clinical people who are keen on knowing the recent most development in therapeutics and pharmacology. Right. As you know, venom is a toxic secretion of an animal which could develop multiple toxicities following an accidental injection to the victim, like human. And hence, venom and the venomous animals are considered as dangerous. That is why they are dangerous. Most of the research have been focused towards the treatment of toxic effect of animal venom. A good example is snake envenoming. Everybody are researching and talking about the clinical effects and their treatment for the snake bites. However, the only few curious scientists have explored the possibilities of use of the animal venom to treat some deadliest and chronic diseases. Numerous such efforts were failed due to many reasons, but few remarkable discoveries are at the practice of clinical medicine, which used to treat some fatal diseases. During the next few minutes, I would explain some great discoveries in medicine that explored some active ingredients of animal venom and their therapeutic use. While the venom were placed in many traditional healing processes during the last millennia, the modern translation of toxins into therapeutics began in 1940. Only few people are aware about the discovery of captopril, the famous angiotensin converting enzyme inhibitor. This potent AC inhibitor was first experimentally demonstrated by the Nobel laureate Sir John Robert Vane in the UK. The structure of ACE inhibitor was dependent on the discovery of a small protein isolated from South American pit viper by a Brazilian pharmacist called Tiago Ferreira. This potent antihypertensive agent captopril is a gift of snake venom, even though you are not aware about that. Even without any other examples, the discovery of AC inhibitors from snake venom would sufficient to justify the therapeutic use of animal venom based on over 1 billion of hypertensive people are currently treating with ACE inhibitors. Venom has evolved at least 88 times in the history of evolution of animal kingdom. As a result, venom is potent defense and a predatory weapon in many animal groups. Sea anemones and jellyfish are considered as the most primitive venomous creatures, followed by spiders, scorpions, centipedes, beetles, ants, and butterflies. Another branch of venomous creatures include corn snails, octopus, squids, starfish, sea urchin, rays, and bonyfish. Snakes, lizards, one bird species, and mammals also. There are over 200,000 venomous animals in the world. Majority of them are arthropods. Most of the venomous animals use their venom for either defense or as a weapon for the predation. Humans are accidental victims of all venomous creatures due to an unexpected animal-human contact. None of the defensive or predatory weapon of animal have evolved against human, because humans have evolved millions of years after the evolution of the other animal groups. This map shows the death rate from the venomous animal contact in the world based on the WHO Global Burdens of Disease 2017. India and the Eastern Papua New Guinea has the highest death rate due to animal contacts. Fortunately, Sri Lankan death rate is below 0.5 per 100,000 population. Most of the Sri Lankan animal contact deaths are due to snake envenoming. Venom is a complex to simple secretion, toxic secretion produces within a specialized venom glands in a venomous animal. And those venom should deliver to the victim or to the prey animal through specialized venom delivery apparatus. As you well know, the fangs of the snake is a specialized venom delivery apparatus, which they deliver the venom to the victim. Single venom composed of few to many dozens of toxic molecules called toxins. Therefore, venom is a mixture of toxins. Some animal venom, such as scorpions, is very simple. As you can see here, only 2 toxins are composed of most of the scorpion venom. But most of the animal venom are complex. Russell's viper venom is composed of over 40 different toxins, and which is one of the most complex venom in the world. That is why still the researchers and the clinicians are struggling for the treatment of Russell's viper bite in Sri Lanka. This is a mixture of 40 different toxins. Every toxins have their special targets in the human body. Venom exhibits two fundamental mode of actions in victim's body. Some toxins act as enzymes that catalyzes a special biological reaction. As an example, all Sri Lankan viper venom contain potent toxic enzymes which directly act on the human clotting cascade. Ultimately, that lead to coagulopathy, bleeding, and other complications. Factor 10 and factor 5 activating enzymes in Russell's viper venom stimulates the conversion of factor 5 and factor 10 into its active form and ultimately activate the whole clotting pathway and lead to consumption of the clotting factors. Similarly, the Sri Lankan soy scale viper venom, we call Valipolanga, activates prothombrine directly, and the enzymatic activity of snake venom, most of the time they accelerate the clotting pathway, and rarely the snake venom can inhibit a specific reaction of the clotting pathway. That is how the animal venom acts through the enzymatic activity. I'm sorry about this because I have converted my presentation into PDF. This video will not be displayed, right? This video will show how one drop of Russell's viper venom accelerate the human clotting cascade. Within 2 seconds, one drop of Russell's viper venom can completely solidify the liquid form of venous blood into solid blood clot. And that is I wanted to show the potency of Russell's viper venom, but that is not working here because my laptop is not possible to connected into this system. The second mode of action of animal venom is through the receptors or through the ligand binding ability of venom, which lead to either activation or the blockage of the specific biological pathway under the regulation of receptors. The best example is coming from Sri Lankan Indian crate venom. Indian crate venom composed of a toxin called Bangaru toxin. This Bangaru toxin is a potent post-synaptic blocker. These Bangaru toxins reversibly block the nicotinic acetylcholine receptors at the neuromuscular junction. And that will completely paralyze the neuromuscular transmission and patient may lead to life-threatening respiratory paralysis. Therefore, basically, there are two main modes of action of animal venom, enzymatic activity and ligand binding activity. Homeostasis is maintained by two main systems of the human body, by nervous system and the endocrine system. Nervous system controls through their neurotransmitters receptors, and the endocrine system act through their receptors and the enzymatic activity at the cellular level. Toxins in animal venom derange the homeostasis through either enzymatic or receptor modulation, and therefore venom is toxic and dangerous to humans. As you know, the pathogenesis of the most of the diseases are due to the fundamental issues at the enzymatic level or the receptor level. Therefore, the reversal of the receptor and the enzymatic activities by the component of the animal venom is possible and justifiable. This is the basis for the exploration of animal venom as a treatment modalities for various diseases, including the discovery of ACE inhibitors for hypertension. In Malaysian Peninsula, there's a small pit viper called Malaysian pit viper. Genetically, this pit viper is very close relative to our hump-nosed vipers. This Malayan pit viper venom is composed of a proton fibrinogenolytic enzyme, which is called viprinix. This viprinix can digest fibrinogen into their fibrinopeptides and lyse the fibrin clot. Therefore, now the discovery of this potent fibrinogenolytic agent from this snake venom is now used to treat DVT, pulmonary embolism, ischemic infarction, and other hypercoagulable status. Even though this drug is not available in the hospital to treat these diseases at the moment, the phase 3 clinical trials are underwent to confirm the clinical safety and the effectiveness. I think you are well aware about tibocurare is one of the key active ingredients in curare isolated from South American poison dart frogs. By binding to the nicotinic acetylcholine receptors at the neuromuscular junction, tibocurare blocks the transmission of excitatory signals from the motor neurons to the skeletal muscles, causing muscular paralysis. Introduction of tibocurare into the anesthetic practice as a selectively acting muscle relaxant improves the practice of anesthesia during last 50 years. Deposition of beta amyloid proteins in the brain is a hallmark feature of Alzheimer's disease, if you can remember your basic pathology. Currently, there are no agents discovered and used to stimulate the degradation of this beta amyloid protein to treat to Alzheimer's disease. Recently, in 2016, one of my friends, a scientist at the Monash University, Australia, explored the toxin from Brazilian viper venom. He demonstrated the one toxin isolated from this snake venom can stimulate the degradation of this beta amyloid protein. Now, the rest of the research is accelerated and underway to develop a drug to treat the Alzheimer's disease. Sometimes you may heard about this drug called Byetta. This is a modern treatment for type 2 diabetes. Enexostide is a glucagon-like peptide 1 receptor agonist, which approved by FDA in 2005 to use as a hypoglycemic agent to treat type 2 diabetes. Once a week subcutaneous injection of this drug was effective enough to lower the plasma glucose level for a week time. It's a longer period, longer acting hypoglycemic agent. This drug was initially discovered from the toxin isolated from the saliva of the venomous lizard called Gila monster in United States. So you can see here in this graph, this is the blood sugar fluctuation after the standard breakfast with and without Byetta. Now this drug is available in the Western Hemisphere to treat type 2 diabetes. Interestingly, some of the patients who had been bitten by this Brazilian wandering spider developed priapism. They developed erectile responses after the bitten by this snake, this spider. Based on that observation, scientists were keen to discover what is in this venom called erectile dysfunction, erectile response of the victim. And following the observation of developing priapism after the spider bite, further experimentation revealed there's a toxin in this venom called TX26. This active toxin acts on different pathways in the endothelium and release nitrous oxide into the vascular smooth muscles and lead into the relaxation of muscular smooth muscles in the vasculature. That lead to that lead to priapism. And now the three groups of scientists in the world working towards whether they can use this TX26 toxin to treat most of the people who are suffering from erectile dysfunction. You may have heard about this drug. It's called pyropheniban. It's a newer generation platelet inhibitor. This is a reversibly antagonize the platelet-platelet interaction and platelet binding to the fibrinogen also through a special receptors called GP2B3A. And these receptors are the fundamentally important receptors to make the platelet clumps at the platelet aggregation process. The results reveal injection of this pyropheniban 90% inhibits the platelet aggregation for a longer period of time. Now, this is already approved and treating for the acute coronary syndrome and post-MI angina. This pyropheniban is isolated from African soy scale viper venom at the the beginning of their discovery. Now, all these drugs are not isolated or extracted from snake venom or animal venom. They can synthesize these molecules at the pharmaceutical laboratory. A close relative of this snake is inhabited in Sri Lanka. Sri Lanka has soy scale viper, genetically very close to this African soy scale viper. But nobody has ever studied about what our soy scale viper venom is composed of. Bivalirudin is a potent direct thrombin inhibitor, originally isolated from medicinal leech saliva. Among very few direct thrombin inhibitors, bivalirudin directly inhibits the action of thrombin and now used as a potent anticoagulant in clinical practice. When you compare the heparin, there are a lot of advantages of bivalirudin because heparin is leading to heparin-induced thrombocytopenia, but bivalirudin doesn't cause heparin-induced kind of thrombocytopenia, and a lot of side effects are less in bivalirudin compared to heparin. And this is a very small, 2-inch size corn snail, a highly venomous animal, live in deep sea ocean. And there's an active ingredient called ziconotide, isolated from this corn snail venom. Now it's available as a drug called ziconotide. It's a synthetic version of small peptide isolated from this corn snail. This potent agent was approved by FDA in 2004 for treating the patients with intractable pain. The compound selectively blocks the N-type calcium ion channels at the dorsal root ganglion of the spinal cord. All the pain-producing pathways which reach to the dorsal ganglion is completely blocked by administration of ziconotide. There's a one drawback of this drug, they have to give only intrathecally because oral or other intramuscular or intravenous administration will quickly degrade this drug by the first pass metabolism and other metabolic steps. Therefore, intrathecal administration is approved for intractable patients by the FDA. The question is whether we can find any anti-cancer agent from snake venom or any other animal venom. It is possible. Snake venom is rich in phospholipase enzymes, which destroys the phospholipid component of the cell membrane. One toxin called protoxin, isolated from venom of the South American rattlesnake. This protoxin specifically selectively go and bind to the abnormal forms of phospholipids in the cancer cells, only in the tumor cells. Selectively act through the cell membrane of the tumor cells and leading to cell destruction and lysis. And this is another advantage compared to the currently using non-specific anti-tumor drugs. What the researchers have shown, increase in concentration of protoxin completely shut down the number of proliferating tumor cells in a cell culture. It's hard to believe some mammals also have some venom. In the history of evolution, some of the mammals have evolved with venom. A little shrew distributed in North America, this is called short-tailed shrew, has venom in their saliva. And shrew use this venom to paralyze their prey animals by blocking the neurotransmission by inhibiting the sodium channels of the prey animal. There's a toxin isolated from this shrew is called soricidine. It inhibits the calcium channels, which is overexpressed in certain tumors like breast, ovarian, and prostate cancers. These cancer cells expresses certain type of calcium channels in their cell membrane. The soricidine specifically and selectively go and block these calcium channels expresses in the tumor cells and leading to malfunctions of all the metabolic pathways of the tumor cells and ultimately induce apoptosis and lysis of the tumor cells. Still, this drug is under the evaluation by different clinical trials before they reach to the clinical practice. This is an example to show how animal venom is rich in bioactive molecule. This table listed various direct thrombin inhibitors can directly inhibit thrombin in the clotting cascade. And they have isolated from various groups of animals, including leeches, bugs, and snakes. But their clinical applications are still underway based on the basic and clinical trials. In my previous slide, I was explaining to you about the Malaysian pit viper. They have a potent fibrinogen enzyme and toxin called Encrot. Now it is under the phase 3 clinical trial to treat for the hypercoagulable status. This is the molecular tree, how genetically close our humpnose viper species. We have 3 species called Hypnale, and Lipa, and Sara. Our humpnose viper species are the closest species for Malaysian pit vipers. Based on our initial experiments, we demonstrated our humpnose viper venom also have this fibrinolytic enzymes. Now, our research group is working towards of isolation of that active component from our humpnose viper venom. Except this work, none of the Sri Lankan venom have been experimentally tested for their pharmaceutical or pharmaceutically active ingredients. But this is the high time to discover these pharmaceutically active compounds from our natural resources like animal venom. As a tropical island, Sri Lanka is rich in venomous animals, including snakes, scorpions, spiders, centipedes, and we have venomous catfish also in our streams. And bees, wasps, and hornets, they are venomous. Except studies on snake bites and snake venom, the effect of other animal venom is lesser known to the medical science. Every venomous animal is a treasure of bioactive molecules. This is the right time to explore these things and how our snake venom and our animal venom work on which enzymes and which receptor levels. In summary, envenoming is due to the disturbances of the homeostasis by the action of toxins of animal venom. Therefore, venom is dangerous and sometimes fatal. Many diseases are due to the alteration of either enzymatic activity or the receptor derangement. Modulation of homeostasis is a key concept of pharmacotherapy. Modulatory effect of animal toxins could be used as a treatment for the diseases, including cancers, coagulation disorders, platelet function disorders, Alzheimer's like long-term neurological disorders, and as analgesics. Hence, the therapeutic potentials of animal venom is obvious. There are hidden treasures of therapeutic agents, but still not explored enough. Therefore, it is worth to investigate about our animal venom, how we can use them as a therapeutic agents. Venom is one of the most diverse and versatile and sophisticated and the deadliest biological adaptation ever to have evolved on Earth. Therefore, venom is described as nature's ultimate weapon. The discovery of pharmacologically active compounds from animal venom would change the understanding of venom, and probably we could redefine the venom as nature's ultimate cure. Thank you. If you have any questions or anything to clarify, I would be happy to answer based on my understanding about this subject. I'm sure this is quite distance from what you are doing at the board setup. Because this is based on basically a discovery of new drugs from our natural resources. But in most of the other countries in the world are moving towards this angle. They try to explore some most of these pharmaceutical active components from natural products and synthetic products. Most of the time when they discover these active components from the animal venom or plant extracts, they don't need to extract and produce this drug from animal venom or the plant extracts. They can synthesize these molecules at the laboratories and optimize the molecular structure, and then testing at the clinical setup and the preclinical setup. As an example, when we discovered a molecule which will have some biological activity, the closely related molecular structures can tested at a given time with the slight modifications. Venom. This is actually, I was working on this hump nose viper venom for last couple of years, and based on the assumption, because we know the Malayan pit viper venom has a potent fibrinolytic enzyme, based on that assumption, I was trying to isolate that similar toxin from Sri Lankan hump nose viper venom, which is there. And what I found, all three hump nose viper venom has very potent fibrinolytic enzymes and compounds. But the problem is the whole venom exhibits the activity. Partially fractionated venom is also exhibit the activity. Now we don't have the facility to isolate that toxin and take it out as a pure compound. Then only we can go into the next step of the clinical trial or the preclinical trial based on that isolated toxin. At the moment, I am actually in the difficulty of that facility, accessing to that facility in the faculty, because we need high standard protein isolation facility for this kind of studies. It's very fibrinolytic enzyme. It's like when we give this compound to the animals, initially we have to artificially make the blood clots, like a hypercoagulable status we have to maintain. And then when we give this drug, which completely lyses the clots, it's like thrombolytic drugs that we use. Still, the fine level of activity is to be discovered. Those facilities are not with me at the moment, but what I can see based on the basic experiment, there are many active components in our animals. Another example is crate venom. Both crate venom, they have potent analgesics because crate bites are painless. Patients describe there's no pain at the bite site. Even with the fang marks, they don't complain any pain. I strongly believe they have potent analgesics, but nobody has ever discovered what's there. No, any other research group is doing this. No, that's the other idea, the intention of disseminating this angle to the medical people and researchers in the country, because it's a natural treasure. It's everywhere. We have venomous catfish, we have venomous sea urchin, venomous corals, venomous spiders, but nobody has ever looked into how they venomous to human and what pathways are going to activate us and blocked.
Australian subcontinent, they have a lot of this.
Australia is home. Yes, there have been, especially the Alzheimer's disease stories from Australia. They are highly keen. More than 10 research groups are working on isolating these pharmaceutically active component from their animal venom. They have the most deadliest snakes, spiders in the world, and they are so potent than our animal venom. And a lot of research groups are working on.
You should probably appreciate kind of work with from our country and
rather than going ahead with green products and find out some way of purifying these things and they use them as therapeutic agents. That's the great thing, and the initiative such should appreciate facilities.
Thank you. And there's the other thing is like the story related to the AC inhibitor discovery. That institute ran for more than 15 years of that single patent. A single patent of the discovery of AC inhibitors was funded for the institute for 15 years without any funds. Now that it's unfortunate our country has no national goal, national or economic goal based on the science and technology based economy. If our country has such goal, like, you can give some targets to the research group, like you have to discover at least 2 drugs within next 10 years. And then those 2 patents would sufficient to run the University of Peradeniya for next 10 years without any government funds. That is how most pharmaceutical agencies and higher education institutes are finding their finances in the Western world. Because one patent is enough. How many pharmaceutical companies will attract and buy that patent for billions of dollars? And that would be enough to run an institute like University of Peradeniya for a couple of years. For that, the government should initiate, and Science and Technology Ministry should initiate the science and technology-based economy. One such angle is discovering new drugs, new therapeutic agents. And there's no such thought even we can see at the moment. Thank you very much.